Tem tantos programas desses, né, que a gente se
Estamos
habitua
lá.
com alguns ou com outros, tem uma curva de aprendizado com cada um deles.
Eu acho que ele desformatou um pouco quando ele converteu. Vamos ver.
É possível.
Já estamos no ar, Hugo.
Okay, so we can start. Good morning to everyone. We arrived to the last day of our event, and we will begin with the pleasure to receive Professor Teresa Cristina. She is a pharmacist colleague, also formed by UFRJ. She has a PhD in biophysics, also from UFRJ. Currently, she's a full professor at the Department of Cell Biology on UERJ. She's a research 1B from CNPq. She is the current head of the graduate program in bioscience at UERJ, and I was quite surprised about the amount of time she is and was coordinator for the program. I counted more than 13 years, Cristina.
About that.
You have
actually we have in between we have some other coordinators, but everybody called me but it's my last year. Next year
you have something like a masochist.
Yes. It's my side, my masochist side.
Well, besides being a full-time coordinator of the program, she acts at the mainly in cellular pharmacology, cellular and molecular pharmacology, particularly work with inflammation, cell signaling and migration. She has around 130 articles. She advised more than 50 people between PhDs and masters, a very large experience in advising students and also postdocs, more than 15 postdocs advised. So a very solid career, both on the scientific perspective and the administrative perspective. And we will have the opportunity to listen a little bit about her work. Cristina, thank you very much for speaking with us and for helping us in this evaluation process. I will be also on the backstage, and the word is all yours.
Okay, thank you, Hugo. It's my pleasure, and it has been very interesting to participate in this meeting to evaluate your graduate program. And it's good for me because I'm cheating a little bit about your experience too, to help our program. So thank you for the invitation. And thank you for the opportunity to show a piece of our work at the Laboratory of Cellular and Molecular Pharmacology at UERJ. And today, we're going to talk about what has been my work, our work in our lab, that's communication in the tumor microenvironment. Actually, our laboratory has a history on the studies on inflammation. I began my career as a researcher, working with inflammation, toxins, etc. And in continuing this experience, we now study the cellular and molecular mechanism involved in the inflammatory microenvironment. I always try to convince people that inflammation is a part of everything of our life. It's not our lives, and it's not a question of defense only, but survival. Survival, any event in your body, since eating to aging, you have the participation of the inflammatory reaction. So our study now is in the inflammatory microenvironment that you can find in many diseases, especially chronic diseases, as cancer and obesity. And when you think about cancer and you look at the landscape of a tumor microenvironment, we understand how complex it is and how inflammation can take a part in this. When you look at this tumor microenvironment, you can see not only this picture shows not only the transformed tumor cells, but also an infinity of other cells, including immune cells, endothelial cells that is going to form the formation of vessels that will feed tumor for its growing, adipocytes, and acellular components like extracellular matrix, microvesicles, and we have studied some parts of these elements, some of these elements. Sorry, but it's a PDF presentation, so some slides are the configuration is not configured quite well. Well, when we study in our group, we have studied the tumor microenvironment in different conditions, and 3 points are the objective of our studies. First, we have been studying the extracellular matrix. The extracellular matrix, that is a very important component of the tumor microenvironment. We studied the extracellular matrix produced by the tumor itself. And you have some works, published works, showing that how this extracellular matrix produced by tumor cells itself can change other components of the microenvironment, like endothelial cells, and we show it in comparing endothelial cell, epithelial cells, normal epithelial cells like melanocytes, and cells from melanoma, human melanoma. These cells produced a matrix, extracellular matrix that, despite the same origin of these cells produced, tumor cells produced different and in composition and also in architecture, the extracellular matrix that can modify the behavior of endothelial cells. And also in tumor that you have heterogeneity of cell types, phenotypes, you can see that some cells can change the behavior of other tumor cells in the same tumor. So I'm going to show it that with the influence of ECM, some cells can undergo epithelial mesenchymal transition and become more metastatic cells. Another point that we have studied is the influence of the adipose tissue on tumor cells. This is a more recent studies that we have showing that adipose tissue you have harvest adipose tissues from obtaining adipose tissue from patients undergoing bariatric surgery, and we observed that this adipose tissue from obese people produce more extracellular vesicles, microvesicles, and these microvesicles can influence, for example, the malignity and the proliferation of breast cancer cells. And so this communication between adipose tissue and tumor cells is one of the focus of our studies. But today I monocratically decided to talk about another element of the tumor microenvironment. There are the tumor-associated macrophages. And we're talking about how different, how distinct can be the metabolism or cellular metabolism of TAMs, tumor-associated macrophages, compared with other phenotypes of macrophages. And we talk mainly about the role or the effects of lipoxin on TAM polarization. This lipoxin is pro-resolution lipid mediators of inflammation, and I'm going to show you what this lipid mediator, pro-resolutive lipid mediator, can change the behavior of tumor macrophage. Well, returning to the tumor microenvironment, as I told you, the tumor microenvironment consisted in an interplay among malignant transformed cells, immune cells, and some other stromal cells, like, as I told, fibroblasts, immune cell, besides immune cells, adipocytes, etc. And it's very interesting how the tumor cells can change this microenvironment, transforming these cells that surround these tumor cells in co-conspirators of tumor growth. So you can, I call your attention about the pro-inflammatory factors that is released by tumor cells that are able to recruit immune cells like macrophages to this microenvironment, and this inflammatory infiltrate can change and co-conspirate, help the tumor growth and development. Tumor-associated macrophages in the tumor cells are the most studied immune cell present in the tumor microenvironment. Is the Hahnemann has called macrophages the accessories to the crime. It's very interesting because these macrophages, most of these macrophages in tumor that can reach about 50% of tumor mass for some in some cancers, most of them are originated from monocytes. The monocytes from bloodstream are recruited towards the tumor. This recruitment is induced by mediators released by tumor itself, and these monocytes-derived macrophages, when they arrive in the tumor microenvironment, they are able to switch their phenotype and transform in tumor-associated macrophages that helps tumor to develop and growth. So it can stimulate tumor promotion. They can regulate the immunity. They can help for inducing metastasis and angiogenesis. And macrophages can do everything of this because they are very plastic cells. The phenotypes and functions that you see, that we can see in that's ruled by macrophages, exclusively depends on the clues, the signals that they receive from the environment. So today, it's well accepted that macrophages can present themselves in two main phenotypes. The 1E1 phenotype macrophage is considered a pro-inflammatory cell with pro-inflammatory behavior, that highly toxicity, and they have anti-tumor activity. These ME1 macrophages are considered inflammatory macrophages because when monocytes are recruited to inflammatory site, finding pro-inflammatory cytokines and bacterial products like lipopolysaccharides, gram-negative bacteria, they change their phenotype to this profile, pro-inflammatory profile. The other profile assumed by macrophages are the M2, considered the M2 macrophages. This is actually a great family of macrophages that present the same markers cell markers, some similar characteristics, but they have, depends on the stimulus they receive, they can assume different functions. They are considered, the M2 macrophages are considered anti-inflammatory cells, and they can participate in tissue remodeling, angiogenesis, immunosuppression, tumor progression. Depends on the environment they found. For example, they are considered, we consider classical M2 macrophages, those that are stimulated in the environment enriched in IL-4 or IL-13. There are other types of M2 macrophages that can participate in immunosuppression or in tissue remodeling that are when they are stimulated by immunocomplex or anti-inflammatory cytokines or growth factors or glucocorticoids. They can participate in tissue remodeling. But there's a special type of M2 macrophages. They are the TAMs, because they switch the profile, the phenotype in contact with the factors released by tumor cells. So monocytes derived from monocytes from bloodstream receive the signal from the tumor that release chemotaxis mediators, and these monocytes can migrate recruited towards the tumor microenvironment, and in contact with this environment, this monocyte-derived macrophage receiving clues from IL-10 and growth factors like TGF-beta, VEGF, microvesicles released by the tumor cells, and even the products from the metabolism of tumor cells. They change their profile to M2 and present some markers that are very characteristic of M2 cells, like markers like CD206, CD163, and enzymes, the expression of some enzymes like a higher expression of arginase 2 and a low expression of nitric oxide, inducible nitric oxide synthase. This is very interesting, these two enzymes, because they share the same substrate, arginine. And M2 macrophages, in contrast with M1 pro-inflammatory macrophages, they present a high expression of arginase 1 and low expression of iNOS. Well, this plasticity of macrophages are very intriguing, because the macrophage polarization into tumor suppressive M1 or tumor promoting M2 involves signaling and changes in metabolic pathways in these macrophages, these cells, but very extremely essential for the resolution of inflammation, but at the same time, it is essential for the establishment of the microenvironment. So these clues, like I told you, can transform M1 macrophages to M2 macrophages. But not only these cytokines and chemokines and growth factor are able to transform M1 macrophages to M2 macrophages and transform these cells in cells that helps the resolution of inflammation or helps tumor growth. Actually, it is, in the inflammatory reactions, the macrophage polarization, the macrophage polarization towards M2 is essential to resolution of inflammation. And many, some specific lipid mediators like lipoxins, resolvin, and myrisins, lipoxins derived from the arachidonic acid, resolvins and myrisins derived from omega-3 fatty acids, can transform switch the N1 profile that produce many able to express and produce chemokines, pro-inflammatory chemokines, produce high amounts of reactive oxygen species and nitric oxide, to switch to Me2 that has present and express, release and express anti-inflammatory chemokines, cytokines, and there is a low production of cytotoxic species. And lipoxin, and today I'm talk a little bit about lipoxins, because lipoxins are it's a biological active mediator that represent a distinct class of arachidonic acid metabolites. Actually, if you remember, the metabolism of arachidonic acid metabolism is mainly during the inflammatory reaction, is mainly driven to the production of prostaglandins and leukotrienes that are lipid pro-inflammatory mediators. But in the course of the inflammatory reaction response, this arachidonic acid can also produce the lipoxin that is a special metabolite, unique metabolite from as the arachidonic acid that is synthesized by several steps, or two or more steps, catalyzed by lipoxygenase. So the lipoxin A4, for example, is synthesized by the action of 15 lipoxygenase followed by 5 lipoxygenase, or 5 lipoxygenase from 2 cells can lead to production of lipoxin. But especially to call attention in lipoxin is that when the inflammatory reaction tried to be stopped by the use of aspirin. Aspirin is a known inhibitor of cyclooxygenase 2 and 1. Aspirin acetylate COX-2, and this acetylation of COX-2 impairs the production of prostaglandins, deviating the synthesis of the metabolism of arachidonic acid towards lipoxin, towards lipoxygenase. And the products of lipoxygenase formed in combination with 5 lipoxygenase present in other cells can produce it on an analog of lipoxin A4 that you call aspirin-triggered lipoxin. That's 15-epilipoxin A4. And our group has been working with a more stable derivative of this compound, lipid compound, that is ATL1. So this is a more stable analog of ATL. Well, this lipoxin, only to tell you, lipoxin and ATL, they act on cells through their binding to a special receptor, ALX. The ALX is a GPCR receptor, and the contact of the binding of lipoxin or ATL to GPCRs can change and lead the cells to lead the organism to a resolution of inflammation. And resolution of inflammation involves the impairing of neutrophil recruitment, impairing or inhibit endothelial activation, stimulates monocytes to migrate towards the inflammatory sites. Since the monocytes migrates transformed, assuming the macrophage phenotype, they can contribute as an M2 macrophage for the resolution of inflammation and other actions that leads to inflammation resolution. Since lipoxin is well known to transform M1 macrophages and M2 macrophages, our group has asked how lipoxin could affect M2 macrophages associated to tumors. Since M2 macrophages are considered a healing, healing of a wound, a healer of wounds, and M2 macrophages involved in the resolution of inflammation, how lipoxin can affect the M2 macrophage attempts. And this is the question raised by Rafael in his postdoc in our lab, and with collaboration of two at that time undergraduate students, Natalia Brito and Hayander. And with this question, reason arose when this question was in our table in front of us, before us, we have to decide how to obtain tumor-associated macrophages. So to obtain this tumor-associated macrophages, we obtain from macrophages originated from human blood monocytes. So human blood monocytes were cultivated for 7 days, and macrophages, they change their phenotype towards a macrophage phenotype, and these macrophages were stimulated with some stimulation, some stimuli to transform these cells in M1 in the presence, M1 with the presence of LPS when interferon, or M2, or classical M2, when they are stimulated with IL-4, or TAMs. To obtain these TAMs, these human macrophages were incubated with the conditional medium of cultures of human melanoma cells. MV3 is a human melanoma cell, highly metastatic. And we use for these experiments, we're going to use in the experiments I show you, ATL1, that's a stable analog of lipoxin. To test, to be sure that our macrophages are really tumor-associated macrophages, had the same characteristic of tumor-associated macrophages, it means it's assuming an M2 profile. We use a cytometry to check if these cells have present M2 markers. So the M2 markers at that time we investigate was MERC1, that is mannose receptor, characteristic of M2 macrophages, and CD163. Two cell markers, M2 cell markers. And we can observe that comparing to non-stimulated macrophages, TAMs, our TAMs, so let me say that as our TAMs present an increase in M2 markers, a sharp increase in M2 markers like mannose receptor, and increase in CD163. And another marker of M2 profile is an increase in an expression of IL-10. So these macrophages have a face these stems have a face of M2 macrophage. They are macrophage, M2 macrophage profile. And how lipoxin would act on this macrophage. Remember that lipoxin is well known to switch the M1 profile to M2 profile. And what you saw at that time, it was a surprise, because when we incubated our TAMs with lipoxin at different concentrations in vitro, we observed that the M2 markers in these cells decrease. The expression of M2 markers decrease, including the IL-10 production. So it was a very big surprise, because these cells have no doubt that they have M2 profile, and how lipoxin can do that. So to test if that lipoxin can do this, they have this effect with other cells, M2 cells. We test in macrophage, non-stimulator or stimulator with different polarized to different profiles. So you can see here when you add lipoxin to M0 macrophage, that's macrophage with non-stimulated macrophages, there are increase in M2 markers. But when you treat these TAMs with lipoxin, as you saw before, I've seen before, there is a decrease in M2 markers. In M1 macrophages, that those stimulated with LPS interferon, that's pro-inflammatory macrophages, there are increase in M2. And classical M2 macrophages, like IL-4 stimulated cells, did not change their M2 markers in the presence of lipoxin. Other stimuli, like TGF-beta in VEGF, are also used as stimulus of macrophages. And TGF-beta in VEGF is factors that has enriched in the conditional medium of the melanoma cells conditional medium. So you can see that cells stimulated with TGF-beta has, in the presence of lipoxin, increased their expression of M2 markers, and VEGF-beta has a decrease, not so very significant, statistically significant, but was a small decrease. But no one had shown the profile of TAMs. So this data pointed us that lipoxin is changing, is interfering in M2 marker, the M2 profile of TAMs, but not with other M2 profiles like a classic M2 profile, classic phenotype induced by IL-4. But were those macrophages, those TAMs, in the presence of lipoxin, they can switch, lipoxin could switch the M2 profile of TAMs into an M1 profile? So to investigate that, we analyzed the no production and expression of INOS and arginase 1. Remember that arginase 1 is an increase in expression of arginase 1 is the hallmark of M2 cells. And we observed that when we take the ratio between INOS, the inducible inosintase, and arginase 1, there is a decrease in this ratio in TAMs. And when we treat these TAMs with a lipoxin, there is an increase there was a decrease and an increase inos arginase 1 ratio in the presence of lipoxin. When we also say the production of NO by these cells, analyzing the metabolites of NO nitric oxide nitrite, and you observe that there is non-stimulated macrophages, TAMs, that has present a low production of NO, and different dose, increasing doses of increasing concentrations of lipoxin, ATL1, increase the production of NO in these cells. And to guarantee that the effect of lipoxin is derived by their action on its own receptor, we use a blocked antagonist of the lipoxin receptor, BOC1, and BOC1 completely inhibit the effect of lipoxin on the switch of macrophage profile, so blocking the production of NO. Besides the production of NO, the macrophage TAMs treated with ATL1 also produces reactive oxygen species. So there is a change in the profile of TAMs in the presence of lipoxin, and these cells can rise the production of ROS, assayed by intracellular probes, and only to TAM, there's no production of ROS. In the presence of lipoxin, these cells begin to produce reactive oxygen species. And reactive oxygen species production together with NO can lead to production of peroxynitrite, and I observed that in the presence of lipoxin, these cells can produce huge amounts of peroxynitrite, and this production of peroxynitrite was inhibited in the presence of antagonist of lipoxin receptor, indicating that the treatment of M2 TAMs with lipoxin can switch the phenotype of these cells towards a M1 profile, M1-like profile, let me say that. And but those species, cytotoxic species, can be effective against tumor cells. And you observe that when we treated macrophages, tumor-associated macrophages with lipoxin, and co-incubate these cells with MEV3 melanoma cells to analyze the apoptotic ratio of these cells. And you observe that in the presence of TAMs, non-treated TAMs, there is a low ratio of apoptotic cells, and the treatment with ATL1 lipoxin increase the number of percentage of apoptotic cells, similar, comparable to those that we observe, that we have observed for M1 macrophages, LPS treated macrophages. And this apoptotic effect of TAM, the TAM treated with ATL, was this effect on apoptotic, the apoptosis induced by ATL treated TAMs was due to the production of the reactive species of nitrogen and oxygen and peroxynitrite, since when inhibit the production of NO or inhibit the production of reactive oxygen loss, we have this effect, the apoptotic effect was inhibited in terms treated with lipoxin. So the data indicated us that macrophages, tumor-associated macrophages, can be a good target for, probably good target for, potentially good target for cancer treatment, since lipoxin can switch the phenotype towards any1 anti-tumoral phenotype. Yeah. But could this effect be also, could this effect seen also in vivo? And for that, at this time, Natalia, Brito, and Hayanda were in their master work, and Rafael continued as a postdoc in our lab. And we decided to treat animals, tumor-bearing mice with lipoxin, to observe the effects of lipoxin and to investigate whether this any effect of lipoxin could be due to its effect, its action on tumor-associated macrophage. For that, our protocol was to inject B16F10 murine melanoma cells in mice, and after the 14 days, these mice present measurable tumors, and at this time, one injection of ATL1 was administered in these animals, and after 21 days, in the day 21, you analyze the tumors. It also took the spleen, bone marrow, and blood. And what we saw is a surprise and good surprise. The treatment with ATL inhibits tumor progression in this incredible way and improve the survival of tumor-bearing mice. So we can see in this graphic that the treatment with ATL, only one dose of ATL, protect animals against death, to death. There is an inhibitor in tumor growth, in volume and weight, and you can see that is a huge inhibition of tumor growth in most of mice. So, interesting it was that early workers that have shown that tumor-associated macrophages can be most derived from monocytes, and these monocytes that originated tumor-associated macrophages are special monocyte phenotype that has an increased expression of LY6CC marker. So what they call pre-TAMs have LY6 are cells, are monocytes with high expression of LY6CC. Is a special population, and they call TREM precursors. But well, when you analyze the non-tumor-bearing mice and tumor-bearing mice, you observe that monocytes of these cells, these animals that are bearing tumor, these cells, these monocytes present a high expression of LY6CC. And what was the effect of lipoxin on these cells? And we observed that in vivo ATL1 treatment decreased the expression of that high expression he decreased the high expression of LY6C monocytes population in all bone marrow and spleen, and also in blood. So, and at the same time, increase LY6C high monocytes with low expression of a population with low expression of LY6C high. So the treatment in vivo with lipoxin can diminish the population of precursors or monocytes that are precursors of TAMs. And what's happened to make sure that these cells could not migrate towards the tumor, you observe that this is CD105 marker for endothelial cells. We observed that we have small tumors and a less expression of diminished angiogenesis. This means when we mark endothelial cells, we are looking for angiogenesis. So there's a diminished the marker, cells labeled with CD105. Diminish the number of macrophages in the tumor. This macrophages in tumor express less arginase 1 and more inos. And you can see here that these cell disease M2 markers and macrophage marker, M2 marker, and you observe it in vivo that after injection of lipoxin at day 14, animals that receive ATL showed a lower expression of M2 macrophages. Yeah. So in this work, we postulate that ITL1, lipoxin analog, diminished the population of LY6 high monocyte in spleen, blood, and bone marrow, decrease in macrophage infiltration to tumor. Lipoxin also reduced M2 marker expression on TAMs, modulating TAM phenotype by shifting M2-like pro-tumor towards an M1 profile. This it induces this change in the macrophages phenotype towards M1, increase the apoptotic effect of this macrophage, decrease angiogenesis, and contribute to impair tumor progression. And our next question is why? Why lipoxin selectively affected M2-like TAMs, promoting a shift to an M1-like profile without affecting M2 macrophages? Why lipoxin affect only TAMs? They did not affect. It did not affect IL-4 M2 macrophages or other types of M2 macrophages. Several questions have risen from this, and more recently we decided to study and publish this work, which is when you asked who would be the selective and specific effect of lipoxin due to differential metabolic profiles of TAMs. As I told you before, in the beginning of this presentation, the shift from M2 to M1 and vice versa, to M1 to M2, requires from cell huge modifications in signaling, in cell signaling, and in metabolic profile of the cells. So could lipoxin to change this metabolic profile? And when we knew from the literature that there is a metabolic differential activation in macrophages that critically support this change in phenotype. This intracellular metabolic change supports this change in macrophage phenotype. It's well known that ME1 macrophages present ME1. Remember, it's macrophage stimulated with pro-inflammatory stimulus. This ME1 macrophage present a high glycolytic flow, a broken Krebs cycle. It means that it deviates the Krebs cycle towards citrates, etc., and a high activity in pentose phosphate pathway to produce more ROS and nitric oxide. In contrast, ME2 macrophages were known to have a high activity in oxidative phosphorylation. It works with intact Krebs cycle and presents a high exosamine pathway that is very important to for the glycosylation of ME2 markers like CD206 and CD301 that are glycoproteins. And exosamine pathway is necessary for this glycosylation. And when you begin to study, to investigate about TAMs in the literature, you can find it's not well elucidated the metabolic profile of TAMs compared to ME1 and ME2. Actually, most works at that time showing TAMs show only its ME2 profile, and there's no comparison to other ME2 profiles. So we decided to return to the beginning and to study how to compare the three phenotypes. So return to our experiments treating human macrophages with ME1 stimulus, LPS interferon, or IL-4, to obtain classic ME2 and TAMs with conditional median of MD3 cells. And comparing the three phenotypes, we observed that ME2 and TAMs present high expression of classical ME2 markers, CD206, CD301, and CD163, while ME1 macrophages present low expression of these markers. ME2 and TAM present lower expression of ME1 markers like CD86. ROS production is low in those phenotypes. Low production of nitrites. ME2 and ME2 and TAM macrophages present a high expression of IL-10 in comparison with ME1, being TAM a production of IL-10 by TAMs higher increase in this expression compared also to M2. Lower expression, genic expression of IL-1 beta, and as a characteristic of ME2 macrophages, ME2, IL-4, and TAMs they present a low phagocytic activity. So how was the metabolic profile of these cells? First, we analyzed the GLUT1, there is a glucose transporter, and we observed that TAMs and ME1 cells have a higher expression of GLUT1 compared with M2. So TAMs are M2 macrophage as phenotype, but it is comparable to ME1 in the expression of GLUT1 that were analyzed by Western blot and also by microscopy. The glycolytic flow in TAMs was higher than M2. The lactate production is higher in TAMs compared to ME1. This is very interesting because early in the literature there is some works showing that the lactate in the tumor microenvironment maintained the ME2 profile of TAMs, and also the TAMs and ME1 macrophages presented a higher expression of hexokinase 2. So until this point, TAMs were more similar to ME1 than to M2. The glycolytic flow, increase in glycolytic flow. Supporting those findings, you also observed that the mTOR AKT signaling pathway that is maintained in there's an increased activity to maintain the glycolytic flow is also increased in TAMs in comparison to M2. But despite that, until this point, the TAMs present a metabolic profile, glycolytic profile, more similar to M1. When we observed the activity of oxidative phosphorylation in these cells, TAMs present a flow of oxidative phosphorylation similar to M2. We have an increase in basal and maximal oxygen consumed, while ME1 is here. ME2 and TAMs presented a higher oxidative phosphorylation, with increased ATP production in comparison to ME1. So TAMs, ME2 TAMs and ME2 IL4 present oxidative phosphorylations profile similar, but in terms of glycolytic flux, TAMs are similar to ME1. So the question at that point was, how important are the metabolic pathways to maintain the ME2 profile of TAMs? Because these ME2 TAMs do not have high production of ROS. They do not have a high expression of INOS, and they do not have most characteristic that is classical of ME1, but they have a metabolic profile similar to ME1. So for this, we use a pharmacological approach, inhibiting those pathways. And when you inhibit glycolysis with the 6-DG, the deoxyglycose, you observe that the maintenance of ME2 profile of TAMs, now you are evaluating the expression of ME2 markers in these cells. Only TAMs lost their ME2 markers in the present when you inhibit the glycolysis. There's no change in M1, no change in M2 cells treated with 6-DG, but TAMs lost their glycolytic, their ME2 expression markers. So inhibition of glycolysis impairs the ME2 profiles in TAMs and the phenotype of ME1. In terms of oxidative phosphorylation, if we inhibit this maintain these cells in hypoxic conditions to observe how the mitochondrial respiration is important to maintain the M2 profile of these stems. We observed that even in hypoxia, when TEMs and M2 did not interfere in the metabolic profile, M2 profile of the stem. Then perform the Warburg effect as well as M1 macrophage. Sorry, but I think that the graphic vanish in this when you convert in PDF. And pentose phosphate, and the pentose phosphate pathway. So if you use inhibitor of the pentose phosphate pathway, we do not restrain in TEMs the expression of M2 markers, but despite when you inhibit pentose phosphate, we lose the expression of M1 markers in M1. The inhibition of pentose phosphate with 6-AN did not interfere in M2 profile of TEMs. So with this work, we state that the results indicate that maintenance of the M2 profile in TEM depends on glycolysis. So TEMs have high glycolysis, high lactate, high fatty acid synthesis that I didn't show you, and have maintained their M2 profile and with inhibition of low expression of ROS, NO, and PPP. And lipoxin could interfere in this lipoxin changing this profile, the stem profile, interfering in their metabolic phenotype. So in a short version, this is not published results. Lipoxin does not affect the glycolytic profile of macrophages. The effect of lipoxin on TEMs does not depend on the glycolytic pathway. So there's not the glycolytic pathway that's essential to maintain the M2 profile of TEMs that is interfered by lipoxin. But ATL tetramet does not alter the oxidative metabolism of TEMs too. However, ATL treatment induced an increase in fatty uptake, and the inhibition of pentose phosphate reversed the effect of lipoxin and the phenotype functional of TEM. So when you inhibit pentose phosphate, inhibition of pentose phosphate reverse the effect of lipoxin in switch the phenotype of stem. You obviously, when you inhibit the pentose phosphate, we also change the functional profile anti-tumoral of lipoxin. Confirming this, when you inhibit pentose phosphate in these cells, lipoxin could act anymore. There's a reverse of the effect of lipoxin, and these cells cannot killing anymore induce apoptosis anymore in tumor cells in experiments similar to that you observed before. Actually, the inhibition of ATL pentose phosphate pathway inhibited the production of ROS and NO, and the cells couldn't induce apoptosis. ATL-treated cells couldn't induce apoptosis of tumor cells. This data, I am finishing my presentation. The data we present indicated that TAMs, although generally considered M2-like macrophage, rely on a glycolytic metabolism to support their function. And the treatment with lipoxin redirects TAM metabolism through a PPP to increase their anti-tumor restrictions. This is a slide that I usually finish to assert that elements that compose to microenvironment could be targets for new complementary therapy in cancer. And this is the people that are working in this published papers, and with collaboration with Dr. Ivan Ross, right? Heitor Paula Neto and Gabriel Doriniello that performed the experiment in seahorse to study oxfox. And as I was very impressive with our interview with the students, I would like to repeat what I have read recently about the Angela Merkel in his farewell address after 16 years in German Chancellor. Whenever scientific insight is denied and conspiracy theories and hatred are spread, we need to resist. Democracy depends on maintaining our trust in facts. This is science. Thank you. I'm sorry about the slides. That's not quite well.
It was great, Cristina. Thank you a lot. Excellent presentation. For some questions, Professor Guido Lenz states that was a fantastic work, and he asks how common is the coexistence of M1 and M2 polarized TAMs in the same tumor?
Actually, when you look at the tumor, you have M1 and M2 macrophage. You have a mixed population. As far as the macrophage that arrive in tumor, they penetrate into the tumor core. They present a higher proportion of M2. You have a higher proportion of M2 macrophages. The change in the use of tentative to transform M2 and M1 macrophages is one of the therapies that we have. We and others are trying to keep for complementary treatment in cancer. So depends on the tumor too. I don't know if anyone will ask about that, but this effect of lipoxin occurs with other tumors. It means other TAMs that we obtained with other type of tumors, you have lipoxin present this effect to change the M2 profile to M1 profile. But for some breast cancer cell lines, we do not have this effect. For breast cancer cell line highly metastatic like MDA MB 231, we do not, lipoxin can have no effect. So depends on the tumor. Okay. I don't know.
I think this relates with his second question about how dynamic is the M1 and M2 interchangeability, and how does this play a role in tumor pathology?
I didn't understand.
How dynamic is the M1, M2 interchangeability, and how does this process play a role in tumor pathology?
Yes, as I said before, you have, depends on the tumor. You have we can have more M1 and more M2 than M1, because in the periphery of the tumor, usually we have more M1 than M2, and and when the growth, the tumor grows, and hypoxic conditions on this tumor can interfere in this. Okay, I don't know. Depends on the tumor. In this tumor, you have no idea if lipoxin can change this.
Okay. Laurindo also congratulates your work, and he mentions that single-cell analysis in other contexts usually shows many other types of macrophages beyond M1 and M2.
Yeah.
And he asks if you have seen this type of work in tumors.
Yeah. Recently, people have made a single cell experiments showing that actually it's funny, because when you think about M2, M2A, M2B, M2C, M2D, and you can see all the alphabet following this. Of course, that, for example, tumor-associated macrophages, they have a different, they have markers of M1 and markers of M2. So they are not M2 classical macrophage. Recently, as I told you, there is I think that is work in cancer metabolism. The authors show that these types of macrophages can appear in tumors. But as I say, we work with only one tumor cell type, and you're trying to do this. I'd like to do a single cell analysis in the tumors of my.
Cristina, thank you again for your presentation.
Thank you.
Excellent, a beautiful cell biology class for the students as well. And well, we will proceed for the next presentation, that is from Hugo Melo.
Hugo, eu vou passar. Sorry, eu vou. I'm going to pass to YouTube. For me, it's better.
Okay. Hugo, you have the rights now to include your presentation or share your screen as you prefer. Oh, well, where is the hair?
Yeah, I cut it like some months ago.
Hugo is a postdoc, is one of our PNPDs. He is a biologist from Universidade Federal do Rio Grande do Norte. He obtained his PhD at the Universidade Federal do Ceará, with a passage through University of Turku in Finland. Do they have plants in Finland?
Yeah, at least in greenhouse.
Not only snow. He is a specialist on plant physiology, has published more than 10 articles, and a solid presence as the first author of these articles, and papers with a solid impact. So he has a solid career and a scientific background to present for us today. Hugo, the word is yours. I will be on backstage.
Okay, thank you. Can you see my presentation, and can you hear me clearly as well?
Yeah.
Okay, good. Well, thank you for the introduction. As Professor Hugo said, my name is Hugo. I am supervised by Professor Jeanette Fett, and I'm a PNPD in the program, PNPD postdoc in the program. And today I'm going to present this work, which is entitled Improving Heat Tolerance of Cultivated Rice, or Rhiza sativa, a possible strategy to face climate change. So the first question is why we are studying rice, and the answer is quite easy, because rice is the world's most important staple food. and will continue to be so in the coming decades. So rice is very important for people. For example, data from 2016, but published in 2019, showed that rice fed 4 billion people, which is more than half of the world population. And it was the staple food for 650 million hungry people, and is grown by 144 million farms families. So it's very important for population in general, and it is also important for the economy because in 2016 it was valued at $206 billion, which is a lot because it's 30% of the world crop value. However, rice production has been threatened by climate change, and this is why we are studying heat stress or high temperature in plants. Heat stress is one of the major concerns for crop yield in terms of climate change, and heat waves, which is like periodic extreme temperatures occurring. Heat waves are becoming more frequent and more intense over the years. So this image shows this very clear. For example, from the 50s to the 80s, the occurrence of extremely cold and extremely hot temperatures are well distributed. So there was no more hot or more cold years, if I can say like that. But nowadays, for example, from 2005 to 2015, it has been showing that the extreme temperatures are going more to the hot side of the graph. So it means that the heat waves are more and more common. And this is why we are studying heat stress in plants. For example, it is estimated that the rice production in Brazil will decrease more than 10% until 2050, and more of this decrease is because of the climate change, so increases in the temperature. So heat stress or the stress caused by high temperatures affects plants on different parts in different ways, and depending on the species and the tissue, the plant tissue, etc., and the intensity of the stress, the duration of the stress, etc. But in general, heat stress causes lower shoot growth to the plants, or induces lower biomass. It impairs the yield and the quality of the plant products, such as seeds or leaves or whatever. It also severely impacts the photosynthesis, the water use efficiency, causes oxidative stress, changes the structure of the plant, changes the carbohydrate metabolism, etc., and etc. So this occurs from roots to leaves, including shoots, grains, seeds, flowers. And so this is why it's very important to study heat stress in plants. And because it's a severe stress to plants, the plants naturally have some mechanisms to avoid the stress, or at least to face the stress. So in this case, it includes several pathways. I'm not going into the details here, but it includes signaling pathways involving hormones. It includes stomatal control, also changes in the carbohydrate metabolism, changes in the antioxidant metabolism as well. And the plants try to use all of these mechanisms to acquire thermotolerance or acclimation. And because of all that I have presented so far, the main aim of our project is to improve Oryza sativa tolerance to high temperatures through genetic transformation. This is actually only a part, of course, of a bigger project. And I'm presenting the two projects I'm more involved with. So these two projects, which are ecophysiological characterization of Oryza wild species and transcriptome meta-analysis of Oryza sativa under heat conditions, will be presented here now, not showing any final conclusion or very detailed results. The idea of my presentation today here is to show what we are doing to improve Oryza sativa tolerance to high temperatures. So we are using these two approaches, and I will divide the presentation in these two projects, which are different approaches. So the first project, the ecophysiological characterization of Oryza wild species, is based on the fact that the Oryza species are adapted to diverse environmental conditions. And the Oryza genus comprises 23 species and 9 recognized genome types. So this means that the domesticated rice or the cultivated rice, which is Oryza sativa, has wide relatives. In all of these species, of course, are in the Oryza genus. So the wide Oryza species represent a source of genes for genetic improvement of rice cultivars. And this is why we are studying Oryza wild species. So these wild species are distributed all over the world, of course, in the tropical and subtropical regions, because it's the habitat of the species, the genus actually. And here you can see the diversity of species in this Oryza group. So this project, the objective of this project is to characterize the climate and soil conditions where wild species naturally grow, aiming to identify interesting Oryza genomes with high tolerance to adverse conditions, such as high temperatures. So the idea of this project was created with two main informations we had. The first one is that we knew about the availability of the database with registered satellite imaging to monitor the climate on Earth, and including temperature, precipitation, radiation, and this kind of data we see every day in the TV, for example. And we knew that these data were registered over several years. So we had a huge amount of data concerning the climate on Earth. And also, we knew the existence of a database informing the coordinate, the geographic coordinate of samples of different wild species, including Oryza. So this database, Genesis, it has a huge register list containing different species, including wild species, as I said, with the exact location on a map. So here, because of this, we tried to combine these two information and overlap the two maps and make a data matching to try to identify the regions where these species usually grow, naturally grow on Earth. So with this kind of approach, we would be able, and we are actually being able, to identify species that usually grows in more cold or heat regions. The species which would be more susceptible or resistant to drought and flood, or high and low temperatures, etc. Because we had this number of variables through the satellite data. And concerning the Genesis database, we had available the localization of 130,000 samples of Oryza plants from different species. And you can see this here in this map that we made, showing the different samples of different Oryza species registered around the world. So here it's not specified what species is each dot, but you can see that it's well covered around the world. So we would expect to find Oryza in different climates or different regions. But again, of course, most of them are in the tropical and subtropical region, because it's the nature of the Oryza genus. So now showing the results of crossing this data only as an example, because we have much more parameters analyzed already, but I'm going to show only the main ones to trying to show the idea of the project. So we have identified the Oryza species that grow in regions with wider temperature ranges and with severe high temperatures, as shown here in these two graphs. So for example, in this first one, we can see, or we can conclude, that Oryza australiensis, Oryza barti, and Oryza quartata are three species of this Oryza genus that usually grows in regions with higher temperature ranges. So this means that they are more susceptible, not susceptible, but their nature is to be to live in regions that suffer more temperature variances. And here we can see that these three species usually also grows in higher temperature regions with the very extreme temperatures close to 40 degrees, for example. So from here we can conclude that Oryza sativa, Oryza barti, and Oryza quartata, for example, are species that maybe can serve as thermotolerant reference for Oryza sativa, and maybe has we can use the genome with, of course, specific genes to improve the thermotolerance in Oryza sativa, which is the cultivated rice. Also, we analyzed other kind of parameters not related to temperature. For example, some of these species seems to be tolerant to other adverse climate conditions, such as drought. So here you can see that Oryza australiensis and Oryza barti usually grows in regions with lower precipitation levels, and in very dried regions. So with this, this is important actually, because we can't see to the temperature adverse condition alone, since the adverse conditions occur together. For example, a region with high temperatures usually also has lower precipitation or high light densities. So we are trying also to look at this data to not to choose only a species that is thermotolerant, but is maybe susceptible to other climate conditions. And more than that, we are also looking for the soil data where these species are found. So this is a very huge database we have. We didn't conclude our analysis yet, but we have this data, and it's they are physical chemical parameters of the soil, and we are doing. Our intention is to do the same to cross the data from the localization and also to the parameters of the soil database, trying to figure out what are the conditions of the soil where those species usually grow. For example, the pH of the soil or the water retention, the nutrients, and etc. So we are not going to have only the preference in terms of climate, but also in terms of soil. So the short-term perspectives for this project is to select the putative most thermotolerant rice-wide species for further studies. And we are already doing this with our Oryza australiensis. And we're going also to perform in vivo experiments with wild Oryza species, in addition to Oryza australiensis and other we had already started, because we had the wild seeds for these plants, so it's not a problem at all for us to make in vivo experiments related to what we will find with this project. And for long-term perspectives, we plan to identify the Oryza species with the highest potential to provide genetic resource for each adverse environmental and soil condition. And we also plan to perform similar analysis with wild species of other important cultures. For example, wild oat, wild millet, barley, wheat, maize, sorghum, beans, soybean, peanut, grape. So we have this data similarly to Oryza. We have already this data. Of course, we're not going to analyze all of these species and these cultures, but we are open for collaboration if any group have the intention to study the wide species of these cultures. So we already have the knowledge to try something else with other cultures. For the project 2, it's a little bit more simpler, I think. It's a transcriptome meta-analysis of heat-treated plants of Oryza sativa. So our objective here is to identify the most differentially expressed genes in cultivated rice under heat conditions. And our strategy was very simple to reach this objective. We performed the data mining with two of the most important platforms to find available data. And as expected, we found microarray data and RNA sequencing data. And we analyzed this data accordingly, depending on the platform they were they were performed. And we found several entries for each of these platforms. We filtered several of the samples because they were not because of redundancies or because the data was not properly registered, or there were maybe some information missed. So in the end of the filtering process, we had 36 studies composing our transcriptome library, composed of 418 samples, and again divided into microarray and RNA-seq. And these are the numbers of each platform. And most importantly, these data were divided into different categories because as a meta-analysis, the experimental conditions were different. So we have data from different genotypes, different treatment densities, different treatment durations, different plant parts, and we of course are taking care of this. We are very aware about how to group this data and how to properly analyze. But the interesting thing is the different genotypes we are working with, because we have tolerant genotypes and susceptible genotypes. tolerant to high temperatures and susceptible to high temperatures. And we will use this to confirm our data, because it's expected that tolerance genotypes would induce genes more, how can I say, more important for thermotolerance in rice plants. So the perspective regarding this project is to identify the most heat-responsive genes and mechanisms in Oryza sativa. We also plan to transform rice plants based on what we will find here. We have already some genes, some putative genes, but we want to finalize the analysis to be sure about what gene we are going to select for the plant transformation. And we plan to suppress or to overexpress this group of genes, or this gene that we're going to select to try to acquire rice plants with higher thermotolerance. And in parallel, we are also organizing or creating a beginner's guide containing protocols for transcriptome analysis. So this is something extra we are doing, because I have been learning from the beginning these methods about analyzing RNA-seq and the microarray. So because of this, I'm trying to organize protocols for me, but we have plans to maybe share this. It's like a protocol for dummies or something like this. So the general perspectives of this project is to improve thermotolerance of rice plants through two different projects, as I said. So the project one, as I said, we will try to do this by using wild rice genetic resource. And in project two, we're going to reach this by suppressing or overexpressing heat-responsive genes of cultivated rice. These two projects, of course, they can work alone. They can bring us important information alone, but if possible, we plan also to combine both of them, the results or the conclusions of both of them, to try to search and investigate potential genes for thermotolerance, which is what we expect from project 2, but in Oryza wild species from hot regions instead of cultivated rice, which is Oryza sativa, and which would be related to project 1. So I just presented what we are currently doing, but I would like also to share with you some works I've been involved with during the last year. So this one was performed with Breno, which was a student of Márcia Magis Pinheiro, Professor Márcia Magis Pinheiro, and this work was also performed with Felipe Marasquim, Professor Felipe Marasquim. And in this work, we investigated the phosphate starvation response in crop roots, but focused on right roots, and it was a meta-analysis and kind of a review. Similar to what we are doing with the meta-analysis I just presented you, but of course focused on phosphate starvation. I recently also published this paper with my colleagues from Finland, from the University of Turku. It's a review about the photosystem 1 inhibition, protection, and signaling. And this one was a project with the colleagues from the Federal University of Ceará, in which we explored the photosynthetic and redox protection in ex vitro tobacco plants under high light conditions. I'm also involved in these 3 projects, which are under submission, or at least in the very final step of the publication, and I hope it they come to be published as soon as possible. And I would like to say thank you very much to my supervisors, Professor Janete Fett, Professor Felipe Kachinevski, and Professor Márcia Pinheiro Magis, for being so attentive and attentions with my work and with me. And also thank you to our graduate researcher in our project, Guilherme Weber, which has been helping us with the meta-analysis project. Also, thank you to the collaborators from the Professor Márcia Magis group, especially to Paloma Menger, which is also PNPD postdoc here in the program, and also thank you to Alexis King. Also, thank you to people from the Delbane's group, which has been working or helping us with the RNA-seq analysis in the meta-analysis. So especially thank you to the Professor Luis Eduardo Delbene and to Beatriz Castro. And thank you to the funding and support agencies and institutes, the university, the program. Thank you to CAPS, CNPq, and to FAPERGS. Thank you. I hope I was able to share what we are doing, and I'm always open to discuss a little bit more or answer questions. Thank you.
Thank you a lot, Hugo. Excellent and beautiful presentation. From your first part, I was with a question in my mind that Professor Diogo Bonato put it in better words than I could. You showed a lot of data on the ecophysiological part of your work, but you haven't mentioned how you are processing this data. Are you done or expecting to do some kind of clustering analysis, machine learning, genetic algorithms? Are you processing this data by hand, or are you using some scripts, R scripts, Python scripts, or something like that?
Well, I'm not using script. I'm doing by hand, very carefully. But as soon as I have the, how can I say, the method well described, I will be able to make new analysis with other species, for example. But no, I'm not a bioinformatics guy, so I try my best, but I'm doing mostly of them manually. I mean, of course, I use some programs to make the life easier for me, but no, I didn't develop any script or anything like this.
Well, you could. I'm proposing to you talk with Diego or with Márcio. You have a lot of nice tools to correlate this data, to select the data more representative, to help you identify the variables that better characterize the desired property. And if you have the data, the tools to analyze them are huge.
Great, thank you,
Professor Ana Maria. Sorry.
Thank you. Just say thank you.
Professor Ana Maria mentioned that thermotolerance is a multifactorial characteristic. In this sense, do you think a specific pathway network analysis is necessary?
No, I don't know. It's very hard to say something concrete about this, because as she said, that it's a that there are several pathways plants use to try to solve the problem with the high temperatures. But usually people look at the stomatal conductance, for example, or oxidative stress is also something that almost always present when a plant is under stress. But specifically for heat stress, maybe checking for the expression of heat shock proteins would be a way to identify the occurrence of heat. But yeah, it's always difficult because plants are very plastic, you know. It depends on the species as well.
complex scenario.
Yeah.
Hugo, thank you very much for your presentation. It was excellent. E até daqui a pouco.